Because we get application from the grant. No, I actually not. I mean, I find words. I was dedicated for in my
How was it? I mean, I guess they had those all last year.
Yeah, so they're like,
Okay, I think we could probably get started now. Everybody's here, looks like lots of people are here. Lovely to see so many people for the first lecture of this term. That's good. And I think probably for a starting lecture we're in for a treat because we've got Professor Nicky Clayton, FRS, today, going to talk to us about the title that you can see in front of you. Now, I think probably most of us know Nicky. For those that perhaps don't, Nicky is an experimental psychologist whose studies of members of the crow family have quite literally shattered assumptions about the cognitive abilities of non-human animals. And through a series of imaginatively designed experiments, she's shown that members, rooks, jackdaws, jays, and New Caledonian crows can plan for the future. They can think ahead of time and reflect on the past as well. So they can do something quite amazing, which we didn't know about just a few years ago, which is engage in mental time travel. And she's also done work which has led us to understand that members of their social groups have minds of their own. So it's almost that they can do theory of mind as well. Now, her most serious, most recent work uses cognitive illusions, akin to magic tricks, if you like, to explore the blind spots in seeing and the roadblocks in thinking. And she's done this in both crows and humans, as well as even extending her scientific study of cognition and behavior to include cephalopods and mollusks, things such as cuttlefish and octopus. In pursuing her interest in the development and evolution of non-verbal cognition, Nicola also worked with young children and non-human apes as well. Her findings have led her to suggest that intelligence in birds and human and non-human primates evolved independently. This is a significant finding. Nicky's a dancer. She's a movement junkie. She's often described as perpetual motion. For those of us that know her would understand that completely. And she explores dance as a form of non-verbal communication in her work. Interestingly, she was the first scientist in residence at the dance company, Rondare. And it's a title that she's had since 2011. And she continues to do an awful lot of work with Mark Baldwin, who actually ran around there for a great many years. I don't think I need to say very much more. We know that Nicky is actually a very important figure in the university here. I've been around the world with her a couple of times, and everywhere we've been, she's always been treated like a rock star, to be quite honest. So perhaps with no more ado, we should invite Nicky to come and talk to us.
Well, thank you, Clive, for that lovely introduction. And welcome, everyone. It's lovely to be back giving Samwill seminars in person. I don't know about you, but I'm a bit zoomed out, so it's really lovely to see so many smiling faces, colleagues, friends, and new people with whom I hope I will become friends. So in this talk, I'm going to argue that the soft-bodied cephalopods, which include the octopus, the cuttlefish, and the squid, possess a suite of behavioural and cognitive attributes that might be comparable to those found in vertebrates. Despite obvious differences in their morphology, and I mean both bodies and brains, their lifestyle and their evolutionary history. I wish that Dr. Alexandra Schnell could be here today. She's the co-author on this talk and has been an inspiration in many of the experiments that I will describe today. But she's been busy in the field. She's working with National Geographic and Disney Plus, filming on Secrets of the Octopus. She's the lead storyteller and producer, and that's coming out on Earth Day in April. And she's also principal investigator on our latest grant. I'm the co-PI, funded by the National Geographic, called Octo Foresight. And I'll tell you a bit more about that later. We just heard only last week that we've got the funding. So the research that I'd like to share with you today is inspired by our previous work on the cognitive capacities of jays and the other corvids that Clive kindly mentioned in his introduction. We often refer to them as feathered apes because when it comes to cognition, corvids seem to be on a par in terms of performance on problem-solving skills with the non-human apes and monkeys. To whet your appetite of the kinds of things we've been able to show in the cuttlefish so far, we've shown that they can remember the what, where and when of past events, something that Tony Dickinson and I coined the term episodic-like memory to suggest that this satisfied the behavioural criteria for episodic memory. And of course, we first showed that in the corvids. And more recently, we've shown that they are also able to exhibit some levels of self-control and future-oriented decisions, such as they eat less crab at lunchtime if they know that their favourite food, shrimp, are served on the menu for dinner. So I'm going to discuss these findings in terms of an evolutionary framework on why these invertebrates may have evolved such cognitive capacities, and also think about some of the implications for what that might mean about our understanding of the evolution of cognition in general, in humans and other non-human animals. So I'm going to start with a few comments about evolution. Now, obviously, this is a myth about evolution. The Scala-Natura doesn't work like a ladder. And my only relief in this particular slide is that the crows, the corvids, are nowhere to be seen on that ladder. I shiver to think where they would put them, probably just above the frogs and just below the lizards. Given that birds and mammals shared a common ancestor over 300 million years ago, a stem amniote, that was just before the split of dinosaurs and crocodiles and lizards. This is a much more accurate representation of evolution, of seeing it as a phylogenetic tree of life, that evolution doesn't work as a ladder. The analogy of a branching tree is much more accurate and more helpful because it gives rise to the possibility that evolution doesn't have to only be homologous. You don't only have to be related to your most close cousins to develop these kinds of cognitive skills. It gives rise to the possibility of convergent evolution or parallel evolution in distantly related species, that share the same, that would be convergent, or similar selection pressures, rather than in homologous evolution. Obviously, it's only based on shared ancestry. Now, in the old days, of course, people considered intelligence or cognition, and I'm going to use those two words synonymously for the purpose of my argument today, to be exclusively the province of the primates. But research over the past 40 or 45 years has shown that much more distantly related species, such as elephants and dolphins, orcas for that matter, but also even more distantly related non-mammalian species, such as corvids, such as the scrub jay and the crow pictured here, and the parrots, such as the African grey parrot pictured here, may also share these cognitive abilities, particularly when it comes to things like evidence for episodic-like memory, the ability to remember the what, where, when of past events. What they have in common, of course, is that they're all highly social. They live to a ripe old age with lengthy developmental periods and therefore lots of opportunity to learn from their siblings, from their peers and from their parents. They have huge brains relative to body size. Indeed, the corvid brain relative to body size is as large as that of the great ape. So they either have very big brains for their bodies or they have very tiny bodies for their brains, depending on which way you want to look at it. And I'm going to just share with you one example for now, which I particularly like. This is one of my favourite corvids, the rook. You see them in colonies around here. Let me just show you this little clip.
Rooks. Closely related to those other birds we've seen so far, crows and ravens, all have big brains. Litter bins. Food. Ravens all have big brains. Litter bins. Food.
Seems to be working properly. It was earlier. Let's see if I can get the crux of the point. So it's really here. What they're doing, despite moving on and playing on this thing, is that they've gone in together to a bin, get crows either side. They're using the bin liner as a tool to pull up the food.
Solution?
And then they toss it over the side. But the interesting thing about this is it takes about 20 knocks to get the food. So it takes time.
Solution?
It's not
Just
simply
watch
just
this.
grab the food and throw it out. And it's teamwork. It takes two to get the food. So it's tools, it's teamwork, and it's time. Now, obviously, when you see an observation in the wild, you don't know anything about the cognitive processes that are controlling that behaviour, but they're interesting observations with which to start investigating empirically. And we've been able to show that they're very good at teamwork and experiments in the lab, that even though they're not natural tool users in the wild, they can spontaneously use tools in the lab if a tool is required to solve a problem. For example, adding stones into a tube of water to raise the water level to get the food, or taking a straight piece of wire and bending it into a huck to retrieve a bucket of food, and so on and so forth. But what of more distantly related animals that lack a backbone? Well, I want to share with you a few invertebrates, and these are just examples, and they're what I think of as potential candidates. I'm not claiming that any of these are evidence of or examples of these cognitive abilities. I'm just saying they might be interesting starting points for those that might be interested in investigating these phenomena in other species.
Rooks.
So here's my first example, spiny lobsters that show migratory patterns in Florida. they have sophisticated navigation skills that they use through the use of a magnetic map, as evidenced by research by Larry Bowles and Kenneth Lohman, published in Science. Bumblebees, this is work from Oli Lukola in Lars Chitka's lab, again, paper published in Science. Here they were trained to use a tool, a squishy ball, to obtain a food reward, and they could even learn by watching other honeybirds do these things. Then there's the common cuttlefish. Well, more about that in a moment when I talk about our own empirical studies. And then there's the coconut octopus. And I'll talk more about that at the very end, but the cool thing about these animals are that they carry a coconut shell. It's a bit like a mobile home. They carry it around with them as a protection. Because obviously one of the biggest problems for most of the living cephalopods, the coleoid cephalopods, is that they've lost their shell. And they're extremely tasty to eat. So they've got the problem that they're living lunch. And we argue that that might be one of the things that has been a selection pressure for their enhanced intelligence. Now, I was originally trained as a zoologist in the other one in Oxford years ago, and the John Krebs, and so I can't help but tell you a little bit, a small bit of evolutionary background before I go into the psychology of these things. So the first thing to say is that here's a phylogenetic tree of the invertebrates, and cephalopods are in the phylum Mollusca. You can see here, and they share this phylum with the gastropods and the bivalve, as you can see in this picture here. And in the middle, you can see the cuttlefish. Now, males have longer outer arms than females. That's one way in which you can tell the difference between the sexes. And all cephalopods have 8 arms, but the cuttlefish and the squid also have 2 tentacles. So should you wish to eat cephalopods for lunch or dinner, don't be fooled. When they offer you octopus tentacles, they're not. They're octopus arms. Maybe they think that calling them tentacles, you'll feel more likely to eat them. Maybe we feel a bit squeamish about eating arms, but I assure you they're arms. What's the difference? Well, the arms have suckers from the tips to the armpits, whereas the tentacles only have suckers on the end, which look kind of club-like, as in the clubs that humans might use in aggressive games, for example. And the other interesting thing about the cephalopods is that in the males, one of the arms, which has the wonderful name of hectocotylus, is modified to be used for transferring the spermatophores to the females. So another way you can tell the difference between the males and the females is that the male cephalopods are much less likely to allow you to inspect their arm. They kind of hold their hectocotylus in great protective devices. Well, that's enough about arms and tentacles. What about their brains? Well, they have a complex nervous system. They have 150 million neurons in the brain, in an octopus, for example, and about 350 million neurons in the peripheral nervous system, the arms, for example. There's about 400 million in each arm. And here you can see an embryonic picture. The yellow bits are the eyes. The greeny bit in the middle here is the brain, with the massive optic lobes, and then the donut shape of the part of the brain in the middle. The purple is the cuttlefish bone. They have about 30 to 35 lobes. And here's another picture of a midsection from Christophe Alves' lab, one of my main collaborators. And here you can see the 2 big optic lobes, the ones shown here by the eye, and then the bit at the top is the vertical lobe, and that's a particular particular interest for many of us because it's thought to play a role similar to the hippocampus in spatial learning and memory. And in fact, many, many years ago, Nick McIntosh was head of department here, as many of you know, and he spent quite a lot of his time early on in his career working in J.Z. Young's laboratory on octopus, looking at spatial learning and memory and reversal learning and all those good things. Now back to behaviour. As Clive mentioned at the beginning, he and Mark often call me the movement junkies. So there's something that I suppose I empathise with, their ability to be so fluid. Of course, they're known for their dynamic camouflage, their cerebellum and their coordination. They can bend and they can shrink in almost any capacity because they lack backbone. That's why I referred to them as soft-bodied at the outset. They can squeeze in the tiniest of places. Now let's hope this movie clip will work. Yeah, I thought he was here a minute ago. I'm sorry about this, they were working beautifully before. What you can see in the movie,
it's trying to pass through is 1/10 the size of its
squeeze
body.
into and out of the tiniest of spaces. So it's one of those things about losing a shell, of course, is that they're just fantastic in terms of their ability to make it difficult for the predators to actually capture them. I really hope we can get this working, because what I wanted to go on to
Octopus
next
can squeeze
is
through
to tell
spaces
you
not even Houdini
about
would
some of the
dare.
work that we've been doing using magic effects. We haven't done any of the magic effects on the cephalopods yet, but we have on humans. So I'll just pause for a minute to see if we can get James to come back in, see if we can show these. But in between, I'll keep talking about what we show. So in the movie clips, Clive was performing magic. I think maybe they're coming back in. Thank you, John. We can't get any of the movies to play, and we don't know why.
James going in, right?
Well, I asked James to help earlier and he was busy.
Ah, right, sorry. So, which movie?
This one here, we've tried two. This is the second one, so it's not just one movie. It seems to be a problem with all the movies. You have the magic touch. Would you mind staying for 2 minutes just to make sure the next will work? Fantastic! What was I doing wrong? I hit the duck. I just want you to see, because I can explain it, but seeing is believing. Just look how they squeeze through. They're all the suckers on the arms.
Octopus can squeeze through spaces not even Houdini would dare.
So there you go. So speaking of Houdini, what I wanted to show you next is something that Clive and I have been doing together with Alexandra Schnell and also my former PhD student, Eli Garcia Pellegrin, using magic effects to look at blind spots in seeing and roadblocks in thinking. And I'll explain to you, once we've seen the movie, why I think this is relevant to the current argument.
Magic tricks are a fascinating window on our minds.
Indeed. Do I have anything behind my ear? I doubt it. Anything in my hand? Of course not. Which means that this should be physically.
They highlight gaps in our perception and attention, which magicians exploit to disguise what's in front of our eyes.
I suppose the coin would be just there. Thank you.
I wasn't expecting to have to deal with real magic in
But
the
what
middle of
about
my
other
Samuel
animals?
seminar.
Do they fall for the same tricks we do? Could their susceptibility to magic tricks highlight flaws in their perception? Or could an ability... Magic tricks are a fascinating window on our minds.
Indeed. Do I have anything behind my ear? I doubt it. Anything in my hand? Of course not. Which means that this
Sorry,
is physically
John.
impossible.
There's just one more movie after this. And then...
Highlight gaps in our perception and attention, which magicians exploit to disguise what's in front of our eyes.
I suppose the coin would be just there. Thank you very much.
But what about other animals? Do they fall for the same tricks we do? Could their susceptibility to magic tricks highlight flaws in their perception? Or could an ability to be misled actually reveal kinds of intelligence we didn't know they had?
How did you become interested in doing magic tricks for animals?
Well, I've always wanted to be a bird. I've always wanted to fly and move around three-dimensionally and think like a bird. So that's how I got into science. But also through my passion for dance, I met Professor Clive Wilkins, who's artist in residence in the Department of Psychology, and he's a professional magician. And we both shared a great interest in memory and mental time travel through different perspectives, art and science.
I visited Cambridge University's Comparative Cognition Lab to see how the group are examining the mental abilities of jays using magic tricks.
Jays are extremely intelligent. We call them the feathered apes because they're on a cognitive par with the chimpanzees. And they're also very attentive. They really watch what you're doing. And so they became the perfect subjects.
Elias Garcia Pellegrin spent months working with the birds in order to prepare for the experiments.
And one of the things that Eli was able to do was to train them to peck a thumb, to ask them which hand they thought concealed the object he'd hidden.
This trick is known as a French drop.
Good job, baby.
The jays don't fall for it, but perhaps that's not surprising. When humans watch the trick, we assume that the left hand is grasping the worm.
Your brain uses shortcuts when you perceive these movements. It just goes, well, what's the most likely thing to happen? Well, the most likely thing to happen is that when this hand grabs an object, the most likely thing is for a completed transfer to occur. And that's where magicians are very good, because they are very good at seeing how to make a movement which is deceptive so alike a real movement that the brain does not notice it. Even though there are particular kinematics that would tell you that this movement is fake, right? Like the tension. But you don't see that because you're just using the shortcuts in perception. However, birds don't need these expectations to survive because they do not have hands.
But the birds do fall for some other tricks.
Have a look. Where is it? Here. You're wrong. It's not.
On my visit, it wasn't just the birds being treated to a show.
Sam, do you remember the £10 note we talked about? Could I borrow it now?
Yes, I can let you tell.
Excellent. Thank you very much. Watch carefully.
When I watch this trick, I'm pleasantly surprised by the increase in my fortunes. But a similar trick was used to make the jays think their favourite snack had been replaced with a different one.
That's a £20 note.
Basically what we did is that we showed Stuka the waxworm in this case, we pretended to deposit it inside the cup, and then we turned the cups around. Then Stuka went in and chose to retrieve the cup where she saw we put the waxworm in. But in doing so, she found that magically the waxworm had transformed into a mealworm. And then we measured how long it took Stuka here, or any of the birds that we tested, to actually ingest, to eat the worm, okay? And we found that when we did this case, a devalue condition, so their first preferred item magically transformed into their less preferred item, they took way longer than when we did an upvalue condition, so their less preferred item in this case transformed into their most preferred item. Think about the magic effect that Clive did for you. Okay, you were happy because a £10 note became a £20 note. That's great, right? But what if Clive takes your 10-pound note and transforms it into a 5-pound note? It's not that happy anymore, right? You're not that willing to take that 5-pound note anymore. You want to give it back. You want Clive to transform it into a 10-pound note again, don't you? That's what happened here.
Are you doing any magic experiments with any other animals besides corvids?
We're hoping to do some magic effects with primates, and we're also excited about exploring some of these ideas with cephalopods, cuttlefish, octopus, squid.
You do get attached to these animals, especially when you spend a long time working with them. You notice their different personalities. Gone are the days of like scientists with white lab coats, not having any feelings for their subjects. We've learned too much about what's going on in their minds to just treat them as just banal subjects. Because these are not banal subjects. These animals can think, they can feel, they understand the past, the present and the future. And I don't see any point for me not to treat them as such, if this makes sense.
So I have to say that it's a hard job to perform a French drop. I'm sure Clive will be telling you more about these things when he gives his talk next Friday in the Samuel Club, which of course is in the evening, not at lunchtime. But for Eli, this task was even more difficult than a normal French drop. You see, a normal French drop, you've got a coin, which doesn't move. Now, it's a lot bigger than a waxworm, but waxworms are squiggly, and to get the jays to do it really well, you need live waxworms, and they wriggle. And Katie, who was then Eli's fiancée, was very, very tolerant, so much so that they're now happily married and living in Singapore, because the number of waxworms that escaped as he was perfecting the choreography of the hands of doing a French drop with a waxworm was quite interesting. But of course, what we were scientifically intrigued in here, not just that the jays were behaved quite differently if the food reward that they were expecting was different from the one they were not expecting. And, you know, if it was a better outcome, they were fine with it. If it was a worse outcome, like the £10 note being turned into a £5 note rather than a £20 note, not only did they often reject the food item, but their crest would come up, they'd turn their back, you know, like cats do when they show you their bottom and in disgust. Well, the jays do the same thing with their white bum. The crest comes up. And in fact, one time when we were filming with a BBC crew, one of the jays, J-Lo, who's particularly feisty, had just had enough, and she went straight for the cameraman and flew off with his glasses. So, you know, you can decide what that means, but I think in Jay terms that was less than pleased. But we were also fascinated with this idea that they seem to only fall for magic effects where hands behaved in the same way as a wing, particularly because you've got to remember all these jays have been hand raised by me and others in our team. And the way in which we hand raise them and feed them is using a pincer grip to put the worms down their beak. But clearly, this action didn't help them interpret what they think they saw and what they think they remember. They'd had plenty of experience of seeing us doing this action, but they don't have these appendages themselves. So what we thought we'd do was to turn to monkeys, because of course monkeys have hands, and you can have monkeys that have opposable thumbs, like capuchin monkeys, and you can have monkeys that just have fingers and thumbs without opposable ones. And that's what we tested next. Now let's see, John, can I finally do this, or do I need your... John, would you mind? Oh, is it working? Oh wow, thank you! Yay! Thank you so much. Thank you. Big thank you, John. this was done at the Shepherd Wildlife Park just nearby. So the idea here then is that the marmosets that don't have opposable thumbs behave like the jays, whereas the capuchins that have opposable thumbs and the squirrel monkeys that kind of have opposable thumbs but not ones that grip properly were fooled by the same effects as the humans. But when we redid the effect, which we called the power grip, using fingers rather than opposable thumbs, the marmosets were fooled too. So it's this idea of embodied knowledge that the kind of morphology, the kind of body you have with wings or hands with opposable thumbs or arms might affect not just the way in which you're able to manipulate an object, but actually the way in which you interpret your perception and your memory. And that's why we're particularly interested in looking in the cephalopods. And Willa and Victor, PhD student and postdoc in my lab, are going to be starting in Plymouth quite soon to look in more detail at the cephalopods using dynamic camouflage and pursuing some of these unrelated issues in more detail then. So what do we mean by complex cognition? Well, in a paper that Nathan, my husband, and I published in Science in 2004, we suggested that you could think of the corvids and the great apes as sharing a common toolkit consisting of 4 principal cognitive traits, which we called causal reasoning, imagination, mental attribution, and mental time travel. And by mental time travel, I mean the ability to remember episodically the what, where, and when of past events, which we call episodic light memory, and aspects of self-control in order to be able to anticipate and plan for the future. And so we thought it might be interesting to at least look in the cephalopods at aspects of mental time travel. In due course, it might be interesting to look at some of the other aspects, but of course it's more complicated because cephalopods are not social and they're not long-lived. And it seems that the ecological pressures guiding their behaviour are probably due to loss of a shell and predation, and probably also the fact that they're monkey movement junkies and so good at movement coordination. And here you just see a rather lovely photograph of Alexandra Schnell. This is during the filming of Secrets of the Octopus, interacting here with a day octopus on Lizard Island. She named it Scarlet because it's got a little scar above her eye, and you can tell it's a female because she doesn't have a hectococculus, and therefore she's willing to let you inspect her arm, although you might get a bite. And of course, further confirmation that she was a female was when the male showed up, and then of course, tragically, a few days later, the female died, because of course that's what they do. They protect their eggs literally with their life. They don't eat during that time, and eventually they go white and float away into oblivion. So that's another thing that's quite different about the mating habits of the cephalopods. It really is a huge cost on the females. So in terms of looking at behavioural evidence, we chose to focus, as I said, on mental time travel, and specifically to look at 3 components. Episodic like memory, so remembering past events in both space and time. Self-control, delaying gratification to overcome immediate temptations and prioritise future needs, which is why self-control is related to future planning. And then anticipating and planning for a future need prior to behavioural execution. So you might consider the total of octopus. Now let's see.
Our understanding of octopus intelligence took another leap forward when in Indonesia in 2009,
Chickens really work.
researchers reported a veined octopus doing something truly remarkable. It was a discovery that would put our understanding of the octopus onto a new level of sentience, occupied by only a few species of animals. Living on the sand flats, they have very few places to hide, but this octopus has a unique solution to the problem. It carries half a coconut shell with it. But it needs to find something else to complete the set. 2 coconut halves. A perfect shelter. What is remarkable about this discovery is how the octopus dismantles its home. Carries it around. Uses it to hide and to ambush prey. And now it's putting it back together again. This octopus is thinking ahead. This is real flexibility. The octopus has to anticipate to carry that awkward object around and then correctly assemble the separate parts to create a single functioning tool. Our understanding of...
So, is it? So that's a key question. Has she, that female coconut octopus that we just saw, optimized her search behavior for shelter through flexible decision-making governed by mental time travel? Is she really planning ahead? Or is there a simpler explanation? For example, they're often ambushed by mantis shrimp. So are they just carrying the shell to deal with a current motivational problem in the here and now? Or are they really doing it with the future in mind, independent of their current motivational state? And that was a point that was first made by Bischoff Curler and mentioned in early writings in the late 90s by Thomas Suddendorf and Mike Corbelis, called the Bischoff-Curler hypothesis, when they argued that animals other than humans are incapable of taking actions for the future that are independent of their current motivational state. So we can be aware that we're hungry now, but we want to do something different in the future, and we can ignore our hunger now for something in the future. Sometimes, not always, not a good idea to go grocery shopping when you're hungry, but minor detail. But the argument was that other animals cannot do so. And so, of course, the idea is that you could have alternative explanations, you could have simpler mechanisms, maybe stimulus, habit, associative learning responses, you could have hardwired responses, you could have complex cognition. And with a diagram like this, although it might look like I'm implying these are either or, I'm not. You can also have an integration of these different components. So you could have multiple components. You could have cognition and associative learning and hardwired behaviours all contributing to a particular problem. So I'm going to go back to what I know, which is food caching. These animals, as you know, they store food, they hide the food, and they do it for a living. And it's a future-oriented behaviour, obviously, because the only point of hiding stuff now, rather than eating it, is in order to gain the reward in the future. So the benefit is that if you hide it now, you may be able to get more food than if you only ate what you could now, because you can eat till you're sated, but you could hide some more for the future. And if your food is only temporarily available, acorns, for example, aren't available all year round, and jays and squirrels really like them, then if you cache them, you can still eat acorns when they're no longer available in nature. And I'm contrasting here squirrels. I supervise a very lovely PhD student's second project on food caching in squirrels. And obviously I've worked extensively on the jays, and the squirrels really are quite different. In their case, caching seems to be largely hardwired and in response to food distribution and the presence of other squirrels. But in jays, we know that it's much more flexible than that. They can remember what they cached where and when. They can anticipate what will happen where and when. And they can deploy flexible information about the future in interesting ways. So, for example, if you give them peanuts, which they like, and worms, which are delicious, they really love them to cache, but they find that whenever they come back to recover their caches, the worms have gone bad, the peanuts are fine, they will learn over trials, as you can see here, to stop caching the worms, even though they're delicious to eat. So they, if you like, that you can think of it as they're responding to a future motivational state. If worms are going to be bad in the future, there's no point caching them now. Here's the more stringent test of the Bischoff-Köhler hypothesis. Give them two equally preferred foods to cache, A and B. It could be peanuts and cat biscuits, for example. And in one group, they're always pre-fed one of those two foods, A or B, for simplicity, obviously I'm only presenting A. And here, of course, the predictions are that they should continue eating and caching the non-pre-fed food type they've eaten, their fill of A, it's B they want. And we call that the same group. The interesting experimental group, of course, is when you pre-feed them a different food at recovery to the one at caching. So A at caching, B at recovery, or B at caching, A at recovery. Because here, they should continue eating the non-pre-fed food type, but actually they should start caching the pre-fed food type, because when they come to recover, it's actually A they're going to want, not B. And you can see that that is indeed what they do in the graph on the left-hand side of the caching. And it's not that they've gone off the food, they continue eating the same amount of the foods in two cases. In another follow-up experiment that I did with my then postdoc PhD student, Lucy Cheek, we did the same thing, but we did this now as a within-subject design. So we had two trays, and we showed that they can plan for two distinct futures. So they know to put the food that they will want at recovery into the correct tray. But what about cephalopods? Well, in the same way as we've done with the jays, you want to identify potential candidates, putatively complex behaviours, in my example, ecologically equivalent to caching. So something that they naturally do in the wild, but you want to be able to do it in a lab setting so you can do the right kind of controls. Obviously, you have to have behavioural criteria because they're non-verbal. There's no point asking them what they remember about the past. You have to do things like tests of whether they can remember the what, where and when of past events. And we need robust controls aimed to differentiate between complex mechanisms, simpler mechanisms, or an interplay between the two. So we focused on three things: the episodic like memory, remembering what happened where and when, self-control, and future planning. So the first thing is identifying a behaviour. Well, luckily their foraging behaviour is quite helpful in terms of becoming a nice model to look at these things, because they have long rest periods, but they're punctuated by brief foraging bouts. And the issue is whether they can adjust their foraging behaviour in response to changing prey conditions. And like the jays, they have their preferences. At the beginning, I mentioned the difference between crab and shrimp. So you could think as crab as a bit like the peanuts and shrimps is a bit like the waxworms. And indeed, it's interesting because actually, although they spend 80% of the time resting, they can't store fat. So they do need to eat every few hours. So they are pretty motivated, and they can take into account not just prey availability, but changing foraging conditions. So I as I said at the beginning, we were able to show that they eat less crab at lunchtime if there are no shrimp are on the menu for dinner, and that's just the results of the experiment that we published in Biology Letters. But what about what, where, when? And can we devise an experiment akin to the jay experiments, where we let them catch worms and peanuts and give them 2 different retention intervals and see whether, in the case of the jays, if the trays come back after a short retention interval, worms are fresh and they should go for the worms. If it's a long retention interval, the worms are decayed, they don't like perished worms, they spit them out, so they should go for the peanuts. Can we do something similar with the cuttlefish? Well, the way in which we tested what, where, when was to use frozen shrimp and live shrimp. And the thing about the cuttlefish is, although they like shrimp, they much prefer live shrimp. To be fair, so do the jays, they much prefer live waxworms rather than dead ones, hence why it took Eli so long with the French drop. So the idea is that they have the opportunity to get a reward either 1 hour or 3 hours after breakfast. After 1 hour, the only option is the sword prawn. After the long delay of 3 hours, they can have the sword prawn or the fresh grass shrimp, and they much prefer grass shrimp. So the idea is that after the short delay, they should go for the sword prawn. After the long delay, they should ignore the sword prawn and go for the grass shrimp. So we have cues up to show them where the items are. So the sword prawn top and the grass shrimp is on your right-hand side. Sorry, I'm not very good spatially. And now you can see an animation that they should go for the grass shrimp, showing that that's what they prefer. So then you have a delayed feeding period. Now you have your 2 icons again. After the 1 hour delay, you have to think, where did I eat this morning and how long ago? So where did I eat? What did I eat? And how long ago? That's the what, where and when. So the what component, obviously there's the 2 foods. So the sword prawn and the grass shrimp, akin to peanuts and worms. Can I get this showing? No, I can't seem to get it to work. John's not here again. He seems to have a magic trick. But you can see here that they massively prefer the grass shrimp over the sword prawn meat. And we looked at young ones and adults. And the where component is simply, can they remember which icon? So there's a black and white triangle cue that's signalled to swim to within 10 centimetres of the location. Is that one working? Yes. Oh, this one's working. So this is a video of a cuttlefish named Bobby who learned to approach the visual cue to receive prey item. She's a female. You might be able to see from the arms. And then the when component obviously is that it's either you can get the sword prawn after an hour, or at the 3 hour one you can get sword prawn or grass shrimp, and you should go for grass shrimp because you much prefer grass shrimp. This is showing at breakfast time. This is just to establish, of course, that they continue to massively prefer the grass shrimp and that they go to the queue. Then after the 1 hour delay, grass shrimp isn't available. No, that one isn't working. Well, they go to the prawn meat because grass shrimp isn't available. After the 3 hour delay, they go for the grass shrimp. So then in the what, where, when, obviously you have Each day the memory is unique because you're using different locations for every test day. And we show that both the subadults and the old adults, they only live to be about 2 years. It's not like a corvid that can live 20, 30 years or so. But we're able to show that both the 9-month-old subadults and the old adults both show episodic light memory. And of course, that's interesting because we're not seeing any age-induced declines. We then looked at delayed gratification. So this is adapted from the Stanford marshmallow test to investigate self-control in children. And so the idea is you can get an immediate reward, but if you're prepared to wait, then you can get a better reward.
You're about to witness a groundbreaking, yet oddly adorable, psychological experiment. This cuttlefish, after days of training, will succeed in a test of temptation originally designed for humans, challenging our understanding of the origins of intelligence. Which is surprising because
cuttlefish are widely different from humans. They live relatively solitary lives. They have 3 hearts. They have a beak like a parrot. They also have little muscles that can extend to make their skin look bumpy. It can change texture and colour within the blink of an eye.
But to survive, cuttlefish seem to need one more trick: mind over matter. This child is taking part in a now famous, similarly adorable experiment.
In the Stanford marshmallow experiment, preschool-age kids are given a single marshmallow. They're told they can eat it immediately, or they could wait 15 minutes for the experimenter to return. If they're able to resist temptation for 15 minutes, they would get a second marshmallow. TV shows and parents have tried the marshmallow experiment on different kinds of kids just to see all the cuteness that happens when a kid is trying to resist a marshmallow.
Many people have recreated this experiment, posting adorable videos of kids displaying specific, interesting behaviors.
Often the children who are able to do the best in the marshmallow experiment are the ones who can distract themselves away from that marshmallow. So often they'll look away or close their eyes, or sing a little song to themselves, or even pretend that that marshmallow is something else. Delayed gratification is the ability to resist the temptation of an immediate pleasure in the service of a long-term, more valuable reward. So the ability to delay gratification usually starts to develop around 3 or 4 years of age. And by age 5, usually most kids are pretty good at this.
This experiment has been adapted to look for self-control in animals.
One really important way we try to understand the evolution of cognition, including the origins of human cognition, is by looking out into the natural world and trying to see how other animals think and solve problems. We tested delayed gratification in primates by giving them a decision between a smaller amount of food they could have right now and a larger amount of food that they had to wait some delay to receive. We found that chimpanzees were willing to wait about 2 minutes in order to get 4 extra grapes.
Up until recently, advanced self-control like this had only been observed in social species such as apes, crows, and parrots.
The social intelligence hypothesis
All right, I'm going to move on because it's stuck again. So the point was that actually the cuttlefish can wait just as long as chimpanzees. And we found that the crows can wait even longer. And perhaps even more interestingly is that we find that there are individual differences in how good they are at waiting or how long they're prepared to wait. And in both cuttlefish and jays, we find that the ones that are able to wait the longest are also better in other cognitive tests, such as reversal learning, for example, and other discrimination tasks, and in the jays, problem-solving tasks, which is interesting because that's the same as what you find in children, that children that can wait longer are often better in other cognitive tests. So what we're going on to now, and I'm not going to say much about it because it's in its very infancy, but obviously what we've not showed so far is future planning. And what we'd like to do is to use this model of the coconut octopus of carrying the shells to try and look at experiments to see whether they really are doing it for the here and now, just to protect themselves from the mantis shrimp they encounter, or whether they're really able to use them to plan for a future motivational need, independent from the ones they're currently doing. So the way we did it in the Jays was we asked whether they can plan for tomorrow's breakfast. So independent of what their current motivational state, can they plan for the future? So we want to do something similar. So just to wrap up, what I've been arguing is that they seem to have some of these abilities that we found in corvids and great apes. And if you look at an evolutionary tree, we've argued that corvids and great apes undergo the same kinds of selection pressures. They're highly social, they're long-lived with long developmental periods, as well as having big brains, and similar kind of ecological pressures, such as being able to use and manufacture tools to get food they wouldn't even otherwise be able to acquire, and also spatio-temporal mapping, keeping track of ripening fruit trees or degrading worms. But what about the coleoid mollusks, the cephalopod mollusks? Well, they've got weak social pressures at best. They're not social, forming long-lived social networks like the corvids and the great apes. They do have ecological pressures, but they're probably quite different ones. Probably the loss of shell relating to this then high predation, this dynamic camouflage, and even in some cases using a tool such as a coconut shell to protect them. So obviously can't imagine that you could think of this really as convergent minds because they're different selection pressures, but it may suggest independent evolution for completely different reasons, different selection pressures. And that might lead us to think about whether there are some general similarities in these kinds of selection pressures, or whether they're actually completely different. And finally, I would say that the research has also had practical impacts which extend to policy. For example, our findings on cephalopod cognition played a crucial role in shaping and changing UK legislation for enhanced welfare, protection of the cephalopods and the decapods, particularly the true crabs, as pictured here on the right. So, in summary, Alex and I would like to argue that this work has challenged preconceptions about complex cognition can evolve only in socially complex vertebrates, given that the cephalopods seem to do things completely differently. And that these raise questions also about the role of dynamic camouflage, the role of this embodied knowledge of the difference between hands and arms and fingers and thumbs, not just on our manual dexterity, but also on how it might affect our perception and our cognition. And these are questions that we're currently investigating, as well as further implications for welfare. I like to think of it as spineless legislation. And more generally, and for me, I suppose as a zoologist come psychologist, but also conservationist, more generally, the way we treat and respect the other animals with whom we share the planet. Thank you.
Well, thank you very much for that, Nicky. That was an exhilarating talk. Lots of very interesting points raised there, and lots of food for thought as well, which I'm sure many of us could apply to our own particular disciplines as well. Are there any questions?
Hi, really interesting. Going back to the bit about the magic and the differences in what animals were formed with the different kinds of French drug processes, does that maybe give evidence towards those other species other than humans having like a Bayesian kind of prior
experience of thumbs, say, then they wouldn't factor that into their perceptions. And so, like, sort of a common Bayesian mechanism in perception between different species. I think you're exactly right. I suspect it does. I think we need to do a lot more empirical work before we can make that conclusion. But I think this whole idea that whether you have opposable thumbs or not should not only, I mean, we know one of the things you still can't get robots to do is hold a cup. And we understand that, you know, it seems obvious that how it affects manual dexterity. But the idea that this is actually affecting your perception and memory, I think, is quite striking. And I agree that I think looking at Bayesian priors is an obvious way to go. After all, if you think about the nuts and bolts of what a magic effect is, it's that you have a memory of what you saw, and you have an expectation of where you think the thing will be it a coin, a note, a worm, or a sponge ball. But you have an expectation, and it's at that point that the magician has something to violate. If you don't have an expectation that is actually wrong because of an assumption you've made, then the magician can't violate it. You know, if Clive does an amazing levitation effect, and I just think that cups are always floating around in midair, then I'm not going to be surprised by it. But I think looking at Bayesian priors would be a great way to go. What I also personally like about it as a comparative person is that I think it opens up lots of experiments that you could do on lots of different animals. You know, it doesn't have to be on a food caching jay, because you're just making items appear and disappear. So in principle, there'd be a lot of different animals one could look at that have a lot of different body parts. It's just, to me, that to start somewhere, to go somewhere completely new after doing the jays and the monkeys, the cephalopods just seem so alien and so intriguing. You know, I can't imagine what it would be like to have 8 arms. I wish I did. Maybe I'd be a bit better at multitasking. There's all this myth about women being better than men. Well, this particular female, anyway, is not very good at multitasking. But who knows? Maybe if I had 8 arms, I could do loads of different things all at once.
You're doing very well with 2 arms, because you've already been described as perpetual motion. What would you be like with 8 arms? Was there another question? Yes.
Yeah.
Yes, Nicky, thank you so much, Nicky. That was fascinating. I was wondering the extent to which, in cephalopods, the current homeostatic state of the individual can be used to map time. Since you said they can't store fat, it may well be the case that their constant energy balance dictates whether they should engage in a particular behavior irrespective of a representation of time having passed.
That's a lovely idea. I think the short answer is we don't know. But I think it would be really lovely to do something about that. You know, I think there's so much we don't know about cephalopods. We know more than we did, thank goodness, so it's worth pursuing. But, you know, they're so completely different. And I mentioned the thing about fat. I kind of hoped you might pick up on it, because I don't know much about it myself. It's not my area. But it seemed to me it's so unusual that they don't store fat. And there must be some really cool questions one could ask about that. So thank you. And I'll talk to you more about it when we chat next week.
I actually kind of have 2 questions, I guess. The first is, obviously, you know, I work more with birds, but I was doing a little bit of kind of preliminary cephalopod research the other day, just kind of going into the stock, because I didn't know how much would be outside of what I understood. And I was looking I saw some interesting information about how cephalopods, specifically octopi, have technically 9 brains, and I don't know how much of that is like what we would technically consider a brain, or if it's more kind of collections of neurons.
It's the nervous system, so that's why I said there are 150 in the brain, 150 million, I mean, and 350 million in the peripheral nervous system. So when they're saying 9, what they mean is the 8 arms plus
That's,
the donut
yeah,
shape
that
brain.
was what made me think about that because you were saying how having the independent arms. And then my other question was, obviously there's been a good amount of research done in octopi and cephalopods, or not cephalopod, I mean cuttlefish. But the other species of cephalopod that I know of at least is the nautilus.
Right.
I know that they kind of have a, I guess I don't know if you could describe it as a shell.
Yeah, so that's why I said the coleode.
Okay.
Cephalopods
So
have
that's,
lost
that's
the
a
shell.
that's like a sub.
They're not coleoids, they're non-coleoids.
Okay, that makes sense then.
So it's just the nautilus that have the shell, and obviously we can't study their behaviour.
Yeah. No, they're
Extinct.
quite rare too.
Yeah.
Another question, yes.
Chris?
Well, thanks, because you make me think out of the box on a Friday midday in between the horrible things I've been doing in the morning. Which, of course, don't involve anything related to university. I love the idea of the idea that you can say, okay, maybe the Jays can project, can do theory of mind, they can sort of do deception.
Mm-hmm.
And I was thinking, is this the relationship of the deception that you could eventually think about with the change in the camouflage?
Yes, I think so. It's early days, but in fact, Willa, where are you, Willa? Wave at the back. Willa's going to start doing stuff on dynamic camouflage in about 3 months, together with work that she and Victor wave, so everybody can see. They are on the acknowledgements, by the way. We'll be doing, and magic is an obvious thing to do. Another thing is to look more closely at what kind of dynamic camouflage they can do and exactly how flexible it is. And another then is to look at whether they can have deception. I mean, presumably it's a completely different kind of deception because they're not social. But so, it's not like Franz de Waal kind of chimpanzee politic friend and foe kind of thing. But still, I mean, if there's a predator coming your way, presumably it would be really good idea to deceive him or her so that you avoid becoming living lunch. And of course, there's all the interesting stuff. I didn't go into it because, there's only so long to speak, but their mating strategies are completely different as well. The whole idea that you're lateralized, I used this in the first contemporary dance work, Comedy of Change, I did with Mark Baldwin, but they're lateralized, so you can have one side saying, hi guys, hi, let's have a party, or fancy a romantic dinner, I'll bring the champagne, you bring the roses, and then you can have this side going, just go away, just, I'm not going to swear, but off. And just, I don't want anything to do with you whatsoever. And so the whole idea that you can have it lateralized with one side giving off one communicative signal and at the very same time, not successively, but simultaneously, another side giving off a completely different signal, I think would make thinking in more detail about these deceptive questions really fascinating. I don't have clear ideas of exactly what yet. That's the kernel of the idea. But hopefully in 3 or 4 years' time, with the help of these guys, I know Willa wants to ask a question in a minute, we'll have some more answers for you.
So it's as if the animals are giving off signals which are akin to communication, aren't they, really?
Yeah, and the way we represented it in the ballet and contemporary dance piece was that they had unitards, and at the beginning they were white on one side and black on the other. And then we had backgrounds that were either white or black, or half white, half black. And then at the end, all of the female dancers were in all black on both sides, and all the male dancers were all white on both sides. Just as it was a very simple representation of both this camouflage and this behavioural nature of change. Yeah.
Any more questions?
Willa had one.
Willa, have you got one?
I was going to add, we actually do know about some deception in cuttlefish. A larger, more dominant male has a display called the flashing zebra display that he often uses when he wants to mate with a female. Sometimes smaller, sleeker males will actually
displayed the pattern as if they're a female to kind of get in and mate with the female under the larger male's like watchful gaze without the larger male even realizing. So there's a lot of work that we can potentially do with this type of deception, and even though they are fairly solitary animals, they do come together at certain points in their life for activities such as mating. So this is a potential research avenue that I think we're all really excited to look into.
Yeah, cool. Thank you, Willa, for that extra addition.
Any more questions? Well, I think we should wrap that up. That's a very interesting hour and a half that we've just had from Professor Nicky